Okay, in yet another example of the remarkable faculty here at CU Boulder, we're going to hear from Sara Sawyer. She's Associate Professor of MCDB right here. She works on the evolution of host-virus interactions, how we adapt to them and how they adapt to us, and the sort of arms race between us. And her title today, which covers a spectrum of different, I would say, RNA viruses, are viruses emerging from the human-animal interface. Thank you, Sara.
Thank you. Thank you for staying to the end. I want to start by just thanking Larry so much for inviting me to speak here today. Thank you. I also wanted to point out that I'm in Tom Check's awesome new Biofrontiers Institute on East Campus. If you don't know about that, you should look into it because there's so many interesting things going on there. With that, I just want to start by saying that we are experiencing a nonstop onslaught of viral diseases emerging from nature. Just to recap some of the highlights from the last 6 years, I'll remind you that in 2012, you started hearing about MERS on NPR. And then in 2014, it was Ebola. In 2015, it was Zika. 2016, we now have, if you don't know, a very scary bird flu circulating in China. And you wouldn't know this because our press apparently doesn't talk about anything but Donald Trump anymore. But there's actually a huge yellow fever virus outbreak going on in Brazil to the extent that 19 million doses of vaccine have just been mobilized in that country. So there is a lot going on, and unfortunately, these viruses are all of the rather severe variety. You can see that many of these viruses, if you get them, you have a third or more probability that you will die from these infections. And by the way, none of these are gone. They're all still lingering, still infecting humans. So this is really just piling up on us at this point. All of these viruses originated in animals, and they made the jump to humans. So in fact, all of the viruses on this slide and many others have their origins in animal viruses. So birds are the reservoirs for influenza and West Nile viruses. These viruses were acquired by humans from primate hosts. People that come into contact with rodents in certain parts of the world get these viruses, and several really important and significant viruses have emerged from bats. Okay, so when viruses evolve the ability to infect a new species, this is called host switching, and when that new species is us, we give this process a special name and we call it zoonosis. Okay, so this whole session has been about microbiology. Maybe you don't care about zoonosis or this process. I hope you do. But if you don't, I just want to point out that there's another process that's highly related, or another important thing. And that's where we take these arrows and we turn them around. And we infect animals with our viruses so that we can study them in the lab. And so animal model organisms for all of these viruses are an absolutely essential part of our drug and vaccine development pipeline. And so the principles apply in both directions, where you're taking a virus and trying to get it to infect and behave in a new species. Okay, so I want to introduce you to the first principles of my field. The first principles, so what we're talking about are viruses of one species infecting another. The first principles are as follows. You have to have opportunity. So species A has to come in contact with species B at a time when species A is shedding virus and able to transmit it, okay? But in most of the examples in the literature, we also think that some level of viral mutation is also required. So what we know is that if you take something like bat viruses, just a big, grind up some bat tissues and take those viruses, if you were to inoculate those into humans, the vast majority would do nothing because these are bat-adapted viruses that don't infect humans for a reason. And so we think that there's usually evolution that's taking place. And then further, once that transmission to humans has taken place, there's some refinement for the spread of this pathogen in its new ecosystem, which is the human population or another animal population. So I want to give you a real example of each of these three so that you really understand what I'm talking about, because this is the foundation of this field, which is really just growing up right now, I would say. Okay, so principle one is opportunity. This is a village called Miliandu, Guinea. And this is the village. If you haven't heard of it, it's okay. There are only 30 houses in this village. But this is the village where the index case for the 2014-2015 Ebola epidemic lived. It was a toddler, and he was the first person known to die in the beginning of this epidemic. The next people to die were his immediate family members, and then the epidemic began. And so you better believe that a lot of epidemiologists descended on this little tiny village after that epidemic began, and they started asking questions. And one of the things that the villagers pointed out to them was this tree that was just a few tens of yards from that toddler's home. And the villagers told these epidemiologists that the village children, including this child, used to play in this tree. You can see it looks really fun, right? Like a tent. And the villagers also said another thing. They said that this tree was also the roosting site for this species of Angolan free-tailed bats. And so you can see, obviously, it doesn't even take an epidemiologist to see that and to wonder if maybe this could have been the epicenter of this horrid epidemic that swept through Africa. So a team, and by the way, that would have been a hugely significant finding because to this day, we have no idea the animal that starts these Ebola epidemics. We've thought it was bats for a long time for many different reasons, but this finding would have been profound because it's never been proven before. So a team was sent there to sample the bats in this tree, and very unfortunately, but perhaps understandably, the villagers had burned the tree down by the time they got there. So we don't know the answer to this question. Okay, principle number 2, viral mutation. Those kids have been playing in that tree for a long time, and those bats had been roosting in that tree for a long time. What was magic about, that December that this epidemic started? So the idea here, I'm going to illustrate it by just taking this imaginary green bat virus and just reminding you that all genomes, but in particular microbial genomes, experience mutations all the time because every time they genomes replicate themselves, including our own, we do it with these enzymes called polymerases, which aren't perfect, and so they make mutations. So there's no such thing as a viral genome. It's more like a swarm of viral genomes because of all these mistakes being made. And the idea is that occasionally a jackpot combination of mutations arises that allows this animal virus to gain a foothold into this new species. Okay, here's a real example of that. This involves the most famous zoonosis of all, the zoonosis of simian immunodeficiency virus from chimpanzees to humans, which gave rise to the HIV-1 epidemic. So this is a protein alignment of the amino acid code of one protein encoded by these two viruses, the matrix protein. And what these researchers noticed, we know this transmission has happened at least 2 times in the last 100 years, giving rise to 2 of the sort of flavors of HIV that now circulate on the globe. And they noticed that both times the same methionine to arginine mutation occurred at the same position in this protein. And further, if you take HIV and passage it back through chimpanzees, that position reverts back to a methionine. So what this tells us is that this is a really significant sort of species-specific mutation in the virus that reflects something that's different about these 2 hosts. And the same is true in flus. If you take flus from birds like wild waterfowl, they will have at certain key positions different mutations than flus taken from mammals. And it's really important to understand what these are because this virus made the jump twice, and it came up with a slightly different solution each time. And one time that it jumped, it gave rise to the flavor of HIV that is called Group M, which has infected 70 million people in the world. And the other time it did it, it gave rise to a flavor called Group N, which has infected less than 20 people. So very different outcomes depending on the viral mutations that are involved. Okay, principle 3. Once you've made the jump, you might not have the sort of strength or ability to move anywhere. If I can't infect anybody else, nobody's really ever going to know about this, quote, epidemic that infected one person. Okay, here's an example we'll go back to the Ebola epidemic now. One of the things that was so special about the 2014-2015 Ebola epidemic was that we had really cutting-edge, high-throughput sequencers on the ground in the middle of Africa, sequencing viral genomes as this epidemic was progressing. And in the retrospective analysis, retrospective meaning this just came out recently, analysis of that data, this is what researchers found. I'll remind you that the very first cases of Ebola that we heard about were in a country called Guinea. And you can see where that virus was 3 months after the start of the epidemic. At that time, in the retrospective sifting through of the data, we saw the first appearance of a mutation in the Ebola virus genome. And that single mutation caused a single amino acid that was an alanine to change to a valine, just one amino acid in the string, in a protein called the glycoprotein. I'll get back to that in a minute. So now we have 2 flavors of Ebola circulating, the previous one and the new one. And you can see that just a few months later, 4 months later, this new flavor is spreading into the countries of Sierra Leone and Liberia. And 1 year later, this new version has really overtaken. And what was really amazing about these papers was that they showed that this measly little change, one change, increased the ability of this virus to get into human cells. And probably most awesomely, they showed it decreased Ebola's ability to get into bat cells. So this was the process of switching hosts, changing from one ecosystem to the other. It's a slow and stepwise process. Okay, so the latter 2 of my principles both involved virus evolution. So to what are viruses adapting during this host switching process? Well, you might immediately think about antibodies and sophisticated pieces of our immune system that are floating around our body, but it's actually much simpler than that. Because most patterns of species tropism, and that jargony term means the species that a virus can infect, these are evident in cells growing in a petri dish where there's no fancy immune system present. There's no circulating antibodies and T cells and B cells and cytotoxic T cells. Instead, we see things like this. HIV is exquisitely human-specific. And that's very unfortunate for all of us, by the way, because it's been quite an ordeal to try to generate good animal models for HIV because this darn virus is so refined to humans. But interestingly, you can recapitulate that at the level of T cells. If I take T cells from humans and T cells from any monkey on the planet, HIV will grow in the human cells and not in the cells. So it usually doesn't have a lot to do with the sophisticated parts of the immune response that you might think it does. And here's the reason. The vast majority of interactions between the host and the virus happen inside of your cells. A virus enters your body, you either breathe it, you eat it, you get it some other way, and it really just floats around until it finds its home. And its home is the cell type, T cells for HIV, that it can go into, open its Trojan horse, and begin to make 1 billion more copies of itself. It's just floating around until that point, okay? Here's a picture. So this is a picture of HIV entering a T cell, undergoing its magic replication process, and then leaving 1 billion times over, making a lot more virions. And this is showing the 400 different human proteins. By the way, HIV has 9 genes and makes 15 proteins itself. But it requires 400 human proteins to replicate itself inside of the T cell. We know that because you can knock down any one of these 400 one at a time and block HIV replication. Okay, so now I'm going to get back to the host switching part. This is where I'm going. Okay, if this is a cell of a human, and I have a similar cell of a bat over here, most of these proteins that the virus needs are going to translate just fine, because these are like important mammalian proteins doing important things, so they're evolutionarily conserved. Like if a virus needed to use a histone, it can probably use a histone of a bat the same way it can use a histone of a human. That's one of the most conserved proteins we know of. So we think that while there are a lot of interactions here, very little of it is relevant to host switching. And thank goodness, because maybe we can find some rules at play in these processes and understand it. So here's the analogy that I like to give for that concept. Imagine that we have this American, and he's going to move to the UK. If he does, he's going to have to learn how to converse with this British gentleman. And for the most part, that won't be a problem because people in England and people in the United States both speak English. So most of the words will translate for this person just fine, with the exception of a few words which will not translate. So these are the words that are going to be the obstacles to this person assimilating to their new environment, just these few. And these are going to be the words that require that this person learn and adapt. And so it is with host switching and zoonosis. The biggest principle here is that the key concept in this field is that while many host proteins are important for virus replication, only a needle in the haystack are relevant to host switching and zoonosis. So we can understand and predict these processes. I know we can, and we've shown that we can. So let me give you an example of this key concept, this needle in the haystack idea. It involves this snake. It's called a Russell's viper. And this snake has, most people believe, nothing to do with the Ebola epidemic. Okay. But a few years ago, some of my colleagues did the following experiment. They took these snake cells, and there was a reason for this. It wasn't totally random. But they took these snake cells growing on a dish here, and you're seeing the nuclei of these cells stained in blue. And they infected these cells with Ebola virus, human Ebola virus. So here's a crazy experiment, right? We're infecting snake cells with a human virus. And the virus glows green. You can see it's not growing. But if you engineer these snake cells to encode just a single human protein called Niemann-Pick 1, the virus now grows just fine, spreads all over this dish. Okay, I have to stop and point out the significance of this to you because humans and snakes are very evolutionarily diverged. Think Jurassic Park. That's the last time we shared an ancestor, right? So the Jurassic era. So and just like HIV, Ebola interacts with many, many host proteins, and they all translate in this enormous evolutionary divide between humans and snakes. They all work for the virus with the exception of just this one. And this is what we call a thin barrier to host switching. And it's really important that we not that we really care about snakes getting Ebola, but we have to find things like this because existing literature tells us that viruses move into new species where the barriers are thin, where it's easiest, where it requires the fewest mutations. Okay. So there's just one genetic barrier here, NPC1. I wanted to point out NPC1, besides being the receptor for Ebola, which I'll talk about in a minute, is your cholesterol receptor. So snakes encode it too. It's just a little different at the sequence level because of the evolutionary divide between humans and snakes. And it's really, this third point is for the aficionados. It's a little bit nuanced, but I love it. It's really important to find the words that don't translate. Because when viruses do jump between these species, these proteins will drive that evolutionary process. These are the obstacles to which the virus is going to adapt. So if you know them, you know the viral variants to look for in nature that have the potential to infect the individuals that you don't want them to infect, like us, not snakes. Okay. So that Niemann-Pick 1, I just want to bring this up. There are some rules emerging in this field, some generalities, I would say, not rules, generalities. Niemann-Pick 1 is a receptor. Almost all viruses utilize some sort of receptor expressed on the surface of human cells. This is usually how they find their home. This is how they find their cell type that they want to find, is that they have a surface protein, finds a particular receptor, and takes them into the correct home. Very often, these are the needles in the haystack that don't translate. And by the way, that example of that magic mutation that Ebola gained during the epidemic as it was spreading through Africa was in the surface protein that allowed it to interact with that Niemann-Pick 1 better than it had before. Okay, I want to contrast that thin barrier to host switching with an example of a thick barrier. By the way, I want to pause here and say I knew I was the last talk of the day, so I designed my talk on the short side. So hang in there with me, and I think we can end early. Okay, so I meant to say that at the beginning. Okay, so this is an example of like the opposite, a thick barrier to host switching. And extremely unfortunately, the example I'm going to give you is from our best and brightest HIV model organism. The model that we use to study HIV, it's called the macaque. And we use this as a model to develop HIV drugs and vaccines. So I told you these receptors are often barriers to host switching. So here we're trying to get a human virus to infect an animal so that we can develop drugs and vaccines. Just like snake Niemann-Pick 1, the receptor that HIV uses also doesn't work in this species. So there's one barrier, but it gets quite a bit thicker than that in this case. These primates also have a really potent immune factor inside their T cells that is very potent at blocking HIV-1. But they also have another one, and another one, and another one, and another one, and another one, another one, another one. So in all, they have 9 total genetic blocks just in their T cells that each by themselves can completely crush HIV, which is why we've never actually been able to infect these monkeys with HIV. Instead, we have to make these chimeric HIV monkey viruses that we study. So the lessons here are that viruses are very unlikely to make jumps like this. And here we're trying to make that jump in the lab. So we make life hard when we try to model human viruses in animals with such thick barriers. Of course, this model was developed 25 years ago, very early in the epidemic, before we knew about any of these molecules and their importance to this virus. Okay, so I wanted to give one brief example from my own research. And given the forum that you had set up here today, I thought I would focus on a question of where can technology be of use in thinking about these problems. So I'm going to talk to you today, just tell you one small scientific story involving a virus called dengue virus. This virus infects almost 400 million people a year. You may not know much about it, but for people that live in this part of the world, it's a part of everyday life. In fact, here's a picture from a grocery store in Puerto Rico, and you can see that they're even exploiting this virus to sell their bug repellent. This virus is a flavivirus, and it's closely related in life cycle and in many other ways to Zika and the yellow fever virus, which I mentioned both at the beginning of the talk. It's spread by a mosquito, shown here. And we have no vaccine for this virus. In a few percentage of cases, people get very violently ill and can die from this virus, which a few percentage of this number is quite a few people, by the way. So critically, not many people think about where dengue virus comes from, not when the NPR talks about it and things like that, but it comes, like many others, from monkeys. We know that Asian and African monkeys have been the donors of these viruses to the human population. But just like HIV, dengue virus is now exquisitely human-specific. And the virus does not reach high titers in any mammals. So yet again, you'll notice a theme. Every time we have this problem, we typically don't have a vaccine. Because if you don't have a good model, it's really hard to develop a vaccine. It's like a bad computer program, garbage in, garbage out, right? That's the saying in computer science, if the model's bad. Okay, so I have to give you one slide of virology in order to explain how we've been trying to apply technology to this. So just hang with me. This is a picture. The dengue virus genome is one piece of RNA. And it translates its RNA into a single giant protein that embeds itself in the membrane of the endoplasmic reticulum. And that giant polyprotein actually has many different important dengue virus proteins in it, and they have to be liberated from each other. So this little piece of the dengue virus polyprotein cuts itself out, forms a protease, and a protease is an enzyme that can cut peptides, proteins. And this protease then cuts at all these red sites and cuts up all the little pieces from each other. A host protease does it on this side, and therefore liberates all the pieces. Okay, but the problem is that it's really difficult to figure out how this protease is knowing where to cut. We know that the protease recognizes 8 amino acids, but if you look at the different places where the arrows are, you can see that there are really different sequences at each of these places. The human eye can't really pick up a pattern here. Like, how does that protease know where to cut? So recently, this is important to think about because recently it's found that this protease, and you can imagine this, also cuts some host proteins in the cell that the dengue virus is replicating in. You can imagine that would be the case because with something this sloppy, if you introduce this to the 30,000 protein human proteome, it's going to find some things that it probably wants to cut. But it turns out that these are extremely important because what we're now realizing is that this virus is completely reprogramming its cellular environment using this protease. For instance, this is this is data from my lab, but this is previously published by others. It's been shown, I know I'm showing a gel at your symposium, I'm sorry. It's been shown that if you take this dengue virus protease, remember we're talking about this protein right here, if you take it in an active version or a deactivated version where a mutation has been made that it's dead, and you express this really important immune protein called STING, which by the way was protecting us from dengue until this protease came along and started cutting it up. So you can see part of the cleavage product here. This is a partial cleavage because we're massively overexpressing this protein in this experiment, but you can see it's going after one of our immune proteins and cutting it up. So these kind of host proteins that the protease is going after are really important to identify. These are potential drug targets. If we could keep dengue from cutting this, our own immune system would protect us from this virus. But also it's important to understanding zoonosis because the primate viruses that we got this from apparently couldn't do this, and they learned how to do it in the human body. And it's also important to develop animal models because here's why. The current animal model for dengue virus is usually the mouse. And the dengue virus protease doesn't cleave the mouse version of STING. So here is one of the major reasons the virus doesn't reach high titers in any other mammal, because those mammals' immune systems are doing what we wish ours was doing. And this is actually true for all other animal models as well. We've never found an animal that looks like us in this regard. So I'll end the virology slide here by saying, I wish we could just scan the 30,000 human proteome and look for this pattern and figure out all the ways dengue is reprogramming the host. But it's too hard to see by eye, and we need to find these host targets. Dengue is difficult to work with, and you can't use knockdown screens in this case because the virus is already knocking down these proteins. So you'd have the same outcome in the presence or absence of the virus. Okay. So my really great MD Ph.D. student, Alex Stabell, came up with a way to address this problem. And I think Alex is here today. And another young graduate student, Allison Gilchrist, is picking this up. Alex went and talked to some students in Robin Dowell's lab, another professor at BioFrontiers who's in bioinformatics, and he decided to take a machine learning approach. Machine learning is a form of artificial intelligence, which we've already heard something about, and it finds patterns in data exactly like this that humans are unable to find. And then it can, after it's learned the rules, classify new data that it sees into cleaved or not cleaved in this case. So there are over 1,000 dengue genomes that have been sequenced. So all those red arrows where the protease cleaves, we know what the motifs are there in 1,000 different examples. So we can use these patterns, and like I said, you can see that the protease doesn't seem to be reading something at the sequence level. It seems to be reading something biochemical, something in the biochemical environment. So we provided a whole bunch of lookup tables. Here's the lookup table for isoelectric point for each one of these amino acids, so that this algorithm would have something to try to find a pattern in. Okay, and so we have this huge 2 sets of training data in our model. We have the positive, all the 8 mers that dengue is known to cleave in these 1,000 genomes, and all the 8 mers everywhere else in the polyprotein that the protease doesn't cleave. And so these are huge, huge training sets, and Alex built this machine learning model, and the way that you test it is as follows. You cut your data in half, and you train your data on one piece, and then scramble up all the remaining pieces and ask the algorithm to sort them into cleaved or not cleaved. And when he did that, you can see that by the time we're including something like 40 or 50% of the data in the training algorithm, it's correctly sorting all the remaining cleavage sites with upwards of 80% accuracy. So based on that encouraging result, we then took the entire human proteome, those 30,000 human proteins, took out the transmembrane domains and cut everything into 8 mers, and used this algorithm, and we predicted about 100 human targets, depending on your statistical cutoff, and started testing them. And here's an example of our test. We have, again, our inactive protease and our active protease. STING is the protein I told you that this thing cleaves. Here are some of our machine learning predictions. Some of them are correct, and we see cleavage. Some of them are not. But we've been testing these, and we have about a 70% success rate so far. So we think that we can really quickly sort of map the whole constellation of how dengue is reshaping these cells by using this, sidestepping the difficult virology with this virus and how difficult it is to work with. And so we think this is a way we could rapidly respond to new viruses like Zika that come onto the scene. And also, this is almost my last slide, there's another interesting outcome of Alex's work. So in the course of this study, we mapped the 8-mer and sting that dengue is cleaving. Here it is right here. And when you align it to the 3 current animal models for dengue virus, you can see a screaming reason why sting is not being cleaved in these species, because they all have different amino acids right at the cleavage site. So once Alex had found this, he searched the whole GenBank database for any mammal anywhere that has the right cleavage site that might be cut by dengue and then harbor dengue at high titers. There are other issues, obviously. And he found exactly 3 species in the whole database that have the right sequence. Sorry, a 4th is orangutan, but it's endangered, so we crossed it off the list. But these 3 are all rodents, and they're all current biomedical research models. So by doing this, we propose that maybe these are just 3 obvious models to try to put dengue, and we might get higher titers and be able to study things like vaccines in a more realistic model. Okay, so we also have a major effort to optimize the HIV animal model, that macaque, in the right way. We got to get rid of the macaque. All right. So in my final few slides, I just want to get a little bit existential on you. Let me check the time. 5:18. Okay. So this is a picture of tent caterpillars. This is an aspen tree in New Mexico that is completely coated with tent caterpillars, and they've defoliated this entire aspen forest. This is a special ecological phenomenon called a population outbreak. And I just took the ecological definition to share with you. A population outbreak is a rapid change in population density over several orders of magnitude, and only a small number of species are known to have outbreaks, some insects and some rodents. This is one. And so ecologists have been studying these for a long time, including this dedicated group who for 25 years has been counting tent caterpillars on a single island in British Columbia. So what you can see here are the number of tents on the island. The caterpillars live in these families in tents, so they can count the tents. They counted the tents on the island over 25 years. Those are the bars. And you can see that there have been 3 population outbreaks. Notice the y-axis, the scale we're on. 3 population outbreaks over 25 years. What ecologists know is that there's a really clear way that nature takes care of population outbreaks, and that's with pathogens. So on the right-hand side, we see the percentage of the tents that are infected with this tent caterpillar virus. And when there's no outbreak, the virus disappears. But as soon as the outbreak occurs, here comes the virus with the black dots until every tent is infected, wiping down these outbreaks one after another. And I had to present this because you have to wonder, are humans an outbreak species? This was actually an idea put forward by David Quammen in the book Spillover, and it really captivated me because nature is throwing these things at us faster than we can respond at this point. So is this us, basically, is this the consequence of us having defoliated our own forest, so to speak? So it's good for me because I get a lot of seminar invitations these days, but it's not good in general. I just wanted to end with one final thought. On my first slide, I had all the viruses that you've heard about on NPR. But if you really want to freak yourself out, subscribe to this database right here called ProMedMail. I'll read this to you. Let's read it now. This is a random pull from a day that I taught my undergraduate class on this topic. So I pulled one of the day's listings off of it. Undiagnosed disease, fatal, Papua New Guinea, request for information. This is a database where doctors talk to each other and veterinarians too. 10 people have died reportedly from an unknown disease in a remote village along the Owen Stanley Range in Papua New Guinea. This has caused panic and a health concern among other villagers and in nearby areas, as the disease causes extreme swelling to various parts of the body and then they die. Public servants who live in Port Moresby were recently in these villages for the Christmas and New Year's festive season and witnessed this disease. In the Fafia local level government of Musa, 10 villagers in Upper Musa along the two main rivers had contracted the disease, causing the middle and lower Musa people to fear for their lives because of the risk of infection. Symptoms are painful swelling, blah, blah, blah. They were fearful that the disease could be waterborne as they were located downstream. Did you hear about this? No, because this is happening 100 times a day on this database. We don't know where the next big one will come from. You can subscribe to it yourself, and you can watch these doctors and veterinarians talking to each other about these epidemics. Remember those 3 principles? Pathogens jump, and usually they just extinguish. It's only the magic events where the right viral mutations occur. Remember the group N HIV that infected 20 people? Most of the time, these things just peter out, but then they don't. So, all right. So our only hope really is to play, my final slide, our only hope is to play defense and offense. My personal view is that we spend all of our time here freaking out about viruses once they've entered the human population, trying to get diagnostics and drugs and vaccines as quickly as we can. But by the way, we really need good animal models and quickly for these 2 things, so we're trying to help in that way. But we also have to play offense. There are a lot of viruses in nature. We have to figure out which viruses are the most likely to emerge, and those thin barrier conduits are where they're going to emerge. And also we have to, if we can find those, we can do really easy things like implement public health measures, like stopping the mining operations in the caves in Africa where the Marburg-infested bats are roosting. If we just had simple information, we could do simple things like that. And with that, I'll just thank, again, Larry for having me here and my lab and everybody for staying till the end of the day. Thank you so much. Thanks.
Thank you, Sara. That was so scary that now everybody's just reflecting.
I think we need those performers back up here a little bit.
Okay, we have somebody back here who I don't think has asked one yet.
Hi.
Just a question on looking at those 8 MERS where the protease was cutting. I mean, obviously proteins have a 3-dimensional structure that they could have had in common without a sequence structure similarity. So did you put that into the whole?
We have some lookup tables that have like size of side chains and things like that. And yes, we've been thinking about that a little bit. You can see the model's not perfect. Yeah.
Yes.
I have a comment and a question. The comment is, I'm so happy to hear you talk about veterinarians and doctors working together, because I remember when West Nile emerged, there was this disjunct between those communities. And my question was, have you ever thought of using the nod mouse, which has its is immunocomprised, so that you can study human systems in the mouse? So.
Yeah, so that's what people do. I really sort of shorted that whole discussion because of the audience I met. Dengue is studied successfully in mice, but you have to abrogate several components of the immune system to do it, which is not ideal when you want to develop vaccines, right? Because a lot of them work through like cell-mediated destruction and stuff. So it's just not ideal. It's not useless, but it's not ideal. Yeah.
I thought your work, in terms of animal models, your work in identifying the other 3 models, the 13-line mouse or what, squirrel, and the chinchilla, and I forget what the other one was, but shows the importance of all the new sequencing that's going on.
Yeah.
And every year we have 10 or 15 or 20 new animals that have had their genome sequenced, and now we can do that more rapidly, and it becomes really important for work like this.
Yeah, I think so. I agree. Yeah. Great.
Hi.
Hi.
Hi. So I've heard that bat viruses are particularly scary because bats have high body temperatures and very good DNA repair. Is that true? Is that something that you know about?
Oh, what a philosophical question you've hit on there. So there are many people that wonder why bats tend to be such terrible donors of these things to humans. And some people even argue about whether that's even statistically true, because bats are this enormous mammalian radiation, so you have to kind of adjust for some things like that. The body temperature thing is a theory about when we get infection, we develop fever, and heat is part of the thing that helps us kill pathogens. So people have this theory that since bats fly all day, they're really hot mammals, and so they kind of harbor, control all these things that then kill everybody else. But to me, I haven't seen any like super nail in the coffin type data to convince me of that hypothesis. Yeah. Yes.
Hi, great talk. I'm going to sleep well tonight. More of a technical question. I see a great value in knowing the host cell proteins that are cleaved by the dengue virus protease. But wouldn't a more obvious target be the dengue virus protease itself?
Yeah, yeah. I mean, of course. Remember, what does your doctor tell you when you go and he says, well, you don't have a bacteria, you have a virus. I don't have any drugs for you. Go home. So we don't have many drugs for viruses, but for instance, for HIV, we have quite a few. There are a couple flu drugs, so there are some examples. And many of those target viral proteins, right? But that doesn't always work because, like I said, we don't have that many virus drugs. So we're just generating like ideas in the landscape of the host-virus interaction here with the two-pronged benefit of understanding how we can design our animal models better and how we may be able to come up with new ideas, new targets for drugs. Yeah.
So thanks for taking my mind off of politics. I feel like I can give up on CNN and just subscribe to ProMed and have new worries. But because of that, I'm kind of curious if you think it might be possible, thinking big now, to identify thin barriers by analyzing viral sequences. So the way you described here, you had to have both the virus in the animal host and the human one to be able to tell where the where the small number of mutations or the easy mutations were likely to occur. But do you think there's some way to identify the viral proteins that haven't jumped yet that have thin barriers?
Yeah. Well, first I'll just say, to put your mind back on politics, one of the bad things is that these viruses almost never emerge in the United States of America. They emerge in other places, and given that we have like no State Department anymore, you're not gonna build a wall to keep these viruses out of the United States. So
sure we will.
Just to bring you back into misery. Okay, so you're welcome. Okay, so can we just do environmental sequencing, look at tons of animal virus genetic material and predict where these thin barriers are? Because of the technological revolution in sequencing, that's exactly what people are doing right now. And they're hitting a wall because they don't know how to do anything predictive with that information. If somebody can plug that break in the pipeline, our knowledge of this field will explode because that's cheap and easy and abundant data that we just need to know how to interpret better. Yeah, I really come at it from the host side, so maybe we'll somehow come in the middle. Yeah.
One last question over here.
I couldn't help but ask a question. When you just talked about these diseases not emerging in the United States, one could say, and maybe I'm wrong here, but one could possibly say that there's a thick sociological or anthropological barrier because there's less contact of human populations in the United States with wild animals, okay, and that there are thinner sociological barriers in countries with rapidly exploding populations in places like sub-Saharan Africa where there are wild animals living in forests or there's closer contact with bats and so forth. And you provide a lot of data to support your theses, but I think one thing that enters in to some extent, maybe from an epidemiological or sociological point of view, are you could say virtual thick barriers in the United States that limits the contact between humans and the animals that may be carrying
So excellent. So that was my first of my 3 principles, opportunity. So you have to have the 2 things coming in contact in the right. I'm a geneticist, so I'll fully admit I focus more on the latter 2, but that is sort of principle number 1, because if 2 things aren't in contact, it really doesn't matter what viral mutations are existing. Yeah.
Well, I think it's time to thank our 4 amazing speakers. We'll see.